Hey, welcome back to Virology. We're going to go more in depth about the different types. So, the group 1 or double-stranded DNA viruses, replication is the same as the eukaryotic virus. There's the semi-conservative replication, the topoisomerase is needed, DNA polymerase is needed, and you make mRNA from double-stranded DNA. So, the double-stranded DNA viruses are pretty lucky. They don't need to carry anything on them. They are going to just use the host enzymes in order to replicate. The only difference is they're going to code their own DNA polymerase. So the herpes virus is one of the examples. The second group is a positive sense single-stranded DNA virus. These will replicate in the nucleus because they're a DNA virus. They use the host DNA-dependent DNA polymerase to convert the viral genome into the double-stranded DNA, making the negative sense DNA. And then from the negative sense template, they make their genome, which is going to be the positive single-stranded DNA. So thanks to that, the first injection of the positive sense single-stranded DNA. That's going to be used to make the mRNA using the host-dependent DNA, the DNA-dependent RNA polymerase to make the viral proteins. I know it's a little confusing, but the positive sense, remember, it doesn't need anything. It can just attach to the ribosomes and start cranking out viral proteins. So that is what it does. It's going to use the host DNA-dependent DNA polymerase to make its negative sense template, and then from there it's going to make the new positive sense replicates. So those are going to be packed in with the viral proteins and make new virions. So the example is parvoviruses. They do that. Negative single-stranded DNA viruses are going to enter the cell, and using the host DNA polymerase, it's going to make positive sense complementary DNA. So the positive sense, remember, attaches to the ribosome, so that is going to be used to make the mRNA for the viral proteins. The positive strand is also used to make more copies of the negative sense genome. So that's the point of the entire virus, to make its own viral proteins and to replicate its genome. The group 3 is going to be a positive sense double-stranded RNA virus. Those are replicating in the cytoplasm because these are RNA viruses. The genome has both positive and negative sense RNA, and they have to carry its machinery. So it carries its RNA-dependent RNA polymerase. So the positive sense RNA are going to make the proteins, and then the positive sense segments are going to be copied using the RNA polymerase to make the negative sense complementary sense strands, which is going to be the template to replicate the positive double-stranded genome. So it's always like back and forth, right? So if it starts with positive sense, it's going to make negative sense DNA, and from there it's going to make the complementary to that negative sense DNA. So it's not an exact copy of the very first positive sense DNA coming in. So with the complementary strand, they're able to make the double-stranded genome. Okay, so a positive single-stranded RNA virus don't enter the nucleus at all because it can only replicate in the cytoplasm. It needs both positive and negative sense in order to go into the nucleus. So the positive sense is going to code for the RNA-dependent RNA polymerase, and the positive sense strands are going to be directly translated onto the ribosome. So these are going to make this using that code in the RNA. And once it replicates, the negative sense genome is then made, and then from there it makes more positive sense RNA. So the RNA strands and proteins are going to assemble and then are going to be budded out or lysed, or the cell's going to lyse. In this case, it's a single-stranded RNA virus, so it's there isn't a need for complementary strands. Okay, so in group 5, there's going to be the negative sense single-stranded RNA virus. These are going to carry its own stuff. It's not a positive strand, so it needs to carry its own stuff. So anything that's a negative sense, it can't attach directly on the ribosome, so it has to carry its own machinery. It makes the positive sense strand, thanks to the negative sense strand, to make the mRNA for the viral proteins, and then from the positive sense template, it's going to make the negative sense RNA. Let's see. The new positive sense RNA is going to make the mRNA for the viral proteins, and then the new negative sense RNA is going to be packaged and lysed out. And in this case, the negative sense single-stranded RNA viruses are going to acquire an envelope from the host membrane. So in group 6, the positive sense RNA virus are going to use both virus and host cell polymerases. So these are going to carry the RNA-dependent polymerase. And these are going to make the viral protein and the negative sense RNA from the positive RNA template. And then the double-stranded DNA is going to be integrated into the host chromosome in order to be a template to make the mRNA and the viral genome. The host DNA-dependent RNA polymerase is going to form the positive sense RNA for the virus. So this one doesn't really need to make a negative sense template. Okay, so group 7 is going to be a partially double-stranded DNA virus, and it partially replicates it's going to replicate using the RNA intermediate. So it's going to use both viral and host cell polymerases to code for their own RNA-dependent DNA polymerase, and then from there it's going to actually replicate its own genome. Okay, so HIV is also a class 6. It's a single-stranded positive RNA, and this one's unique because it uses reverse transcriptase to make the negative RNA. So using the negative sense RNA, it's going to make positive RNA in order to make the double-stranded DNA in order to be incorporated into the host genome. So thanks to this reverse transcriptase, it's able to make a complement of its negative RNA, and then it goes into the host genome. The host genome does its own thing and synthesizes the HIV proteins and replicates the genome for the virus. The site of the assembly depends on the site of replication, so whether it's in the cytoplasm or the nucleus. In the nucleocapsid, the procapsid is going to be made first, and then it's going to be filled with the genome, or the capsid protein can be assembled around the genome. So a naked virus is going to be released by cell lysis or exocytosis, and the enveloped viruses will leave by budding, because you need to bud in order to get the host membrane. Okay, so that's it for now. Thank you for your attention, and good luck studying.